So listeners note, no camping in the Keys in August.
Science.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am Kim Klonowski.
And I'm David Peterson, your host for today's episode. With the increasing concern about diseases of wildlife and potential spillover to humans, wouldn't it be cool if we had a CDC for wildlife diseases?
Yes, it would. Our guest today is Dr. Mark Reuter, faculty in the Department of Population Health and director of SCWDS, the Southeastern Cooperative Wildlife Disease Study.
So, Mark, the first thing that we like to do is ask folks about how they got into science. So you can start wherever you want, college,
Okay.
kindergarten.
Yeah, it's got to start with some of my first memories, actually. My grandma, I grew up in Kansas. My grandmother had a farm in rural Kansas, and that's really where we spent our weekends. And so my childhood was filled with basically weekends running around on that farm, exploring the natural world, being fascinated with really everything I saw. And my grandmother and my mother really sort of helped sort of feed that curiosity and sort of passion that was kind of burning inside. And it kind of just never went away. And, I went to undergrad as a organismal biology major, and then found myself going to veterinary school, not really realizing at that time what that meant or the diversity of options that it provides for you. And so when I went into vet school, I figured, well, I'll just be a dog and cat vet, a cow vet, and just volunteer with, in the wildlife field and try to contribute to conservation. But once I got there, I realized there's this whole other world of veterinary medicine that I can sort of follow that passion for wildlife conservation and population health. And so yeah, it was all started though with my grandma's farm and collecting bones from dead wildlife and artifacts that I still have to this day. If we were in my office right now, you could see all my bones that I've collected forever.
That's awesome. So just real quickly, talk to me about your mom and your grandmother. Tell me about how they kind of encouraged that. Were they scientists themselves or just ones that let you be free?
Yeah, certainly let me be free. And my grandmother, she was, she was a anthropologist. And so she actually lived on Papago Native American reservation in Arizona for a while, immersive, research. And I think that probably had a big impact on her as far as her appreciation for the natural world, and also just a rural Kansas farmer, so cattle and soybeans and sorghum and stuff, but also very in tune with wildlife on the property and making sure that she was sort of a steward for the land and for the natural world while still producing crops and cattle. And yeah, so that really was, it was pretty cool to sort of have that, as your childhood exploring things. And then I was really fortunate I grew up in Wichita, Kansas, and they had a magnet school system. So it was a public school, but I transferred by myself in third grade to an environmental magnet school in Wichita. And that was one that they had in the schoolyard, different ecosystems represented and habitat types. And it was really cool.
That does sound really cool.
Yeah, and I'm not trying to date you or anything, but thinking about a school that's focused on the environment, it must have been really groundbreaking at its time.
Yeah, it was cool. It's still there to this day. I'd love to go back at some point and see how it's changed. But it was great.
Yeah, you should. So in our opening, we talked about the fact that you are part of SCWDS. Can you tell us about the history of SCWDS?
Mm-hmm. It's a long one, but I'll abbreviate it. So, today, a lot of people are tuned into wildlife health and wildlife population health. We hear about things a lot, even in popular media, that might give us the idea that, caring for wildlife is something, and understanding diseases in populations might be something we need to be concerned about. If we rewind to 1950, it was a very different landscape. This is a time where state wildlife management agencies, so in Georgia, for instance, the Georgia Department of Natural Resources, these are the state agencies. Each state has a wildlife management agency that really is tasked with managing wildlife populations. And so at that time in the 50s, there's really no capacity for understanding disease. These are just, this is wildlife biology. You're managing populations, setting hunting regulations, conserving habitat. But when things die, there's not a lot of capacity as far as diagnostics and just even expertise to understand, okay, why are these things dying? What does it mean for the population? Should the cattle farmer be concerned about it? Should people with dogs be concerned about it? Should people be concerned about it for their own health? And so the Southeastern State Wildlife Agencies, so this was the Southeastern Association of Fish and Wildlife Agencies. This is sort of like the governing body, sort of the club of all of the state wildlife agencies. And so they had the foresight to recognize that, okay, we need to build some capacity, some expertise, and some understanding. And really the thing that started it was large-scale die-offs in white-tailed deer, often around waterways. And this might not seem like that big of a deal today because we're fortunate to have pretty healthy, robust populations of white-tailed deer, maybe a little too robust sometimes.
Not sure if anyone's seen any, but maybe they have.
But if you rewind to the early 1900s, like you see a white-tailed deer, that's like a big deal, right? Like just even see one, let alone harvest one, right? So after European settlement and market hunting of white-tailed deer, just unregulated hunting, significant land use changes, deforestation, lots of ways that we negatively impacted the environment. White-tailed deer became extirpated throughout much of the region. So at that time, white-tailed deer in the Southeast were really kind of clinging to isolated pockets of habitat that were just challenging for people to get to, such as the swampy areas of the coastal plain, and then the more high elevation or sort of difficult to get to areas of like the Appalachian Mountains. And so we still had white-tailed deer, but they were not this sort of contiguous population across the Southeast. And so agencies, all the Southeastern agencies at that time, wildlife management agencies, were actively restoring deer populations. This meant they were reintroducing deer into previous areas. And so if you can imagine the amount of effort it is to capture deer from one area, transport them hundreds of miles and release them into another habitat, from a wildlife manager's perspective, you want those animals that you've put all that investment into to have the absolute best chance of survival because of all that's at stake. And so there was a lot of concern at this time because during all of these efforts, there were these cyclical, meaning every handful of years, massive mortality events that would just kill dozens to hundreds of deer. And so in the 50s, there was a couple of these outbreaks. One of them was centered on North Carolina, Tennessee, Georgia, multi-state outbreak that killed a lot of deer, often along waterways. It was a mystery at the time. Nobody knew what it was, why it just came every few years. And then there was another event a couple of years later. And so the agencies in the Southeast said, okay, well, let's start a cooperative. We don't individually have the resources to hire a veterinarian or a disease biologist, but if we pool our resources, we can get one for the region, and this person or these people can help to better understand diseases of deer, in particular this disease of deer, which we know today to be a disease we call hemorrhagic disease. It's caused by a couple of different viruses that are closely related, and it's transmitted by an insect vector. And we can circle back to that if we need to. But so the agency stood up. The SCWDS is what we call it, but it actually wasn't called SCWDS at first. It was the Southeastern Cooperative Deer Disease Study because it was 100% focused on white-tailed deer.
Interesting.
Yeah. And then a few years later, they realized, holy cow, we're learning a lot about deer, and we still have all these needs with wild turkey, with bobwhite quail, with ducks, all the species that are native to the Southeast. So they changed it pretty quickly. By 1960, it became the Southeastern Cooperative Wildlife Disease Study, which I keep saying SCWDS because that's the acronym, and it's just a mouthful to say the rest. So,
it is.
If you hear me,
But it rolls off the tongue so nicely, though.
I think you're lucky because as a member of the Center for Tropical and Emerging Global Diseases, that is totally unpronounceable as an acronym. So
tough.
SCWDS sounds great.
Yeah. A lot of people are confused though sometimes. So what do you guys have to do with squids? But we really don't have anything
Oh,
to
I
do with
didn't think
squids.
about that.
Yeah. So yeah, I'm really fortunate, and everybody at SCWDS in 2025 is really fortunate to be part of this like incredibly rich history that's approaching 70 years here before we know it. And there's been just some moguls of people coming through that place and mountains of information regarding diseases of wildlife populations in the Southeast. It's a pretty cool spot.
So I want to circle on back to the hemorrhagic diseases. And I guess my first question is, does that just affect deer populations, or does it sometimes cross over into other animals like cows or?
Yep. It's a good question. It certainly can. I guess if every disease sort of needs a poster child, and I'd say white-tailed deer are kind of the poster child for this disease, but it's actually two different viruses, epizootic hemorrhagic disease virus and blue tongue virus, very closely related viruses, but it's actually two distinct viruses that cause disease individually, but they're so indistinguishable when we're looking at the clinical signs it causes or the lesions, what we can see in tissues that it causes, that we kind of lump it into a syndrome, hemorrhagic disease. But it does affect more than just white-tailed deer. In the wild cervid or in the wildlife arena, mule deer are also susceptible, but a little less so than white-tailed deer. Bighorn sheep, pronghorn, elk to a much lesser degree, bison to a much lesser degree. And then on the domestic animal side, cattle certainly can become infected with these viruses and quite routinely do. Most often they aren't too bothered by it. A lot of times it's more of an inapparent infection. It can impact them clinically and cause some disease. And there's been outbreaks in other parts of the world that have been pretty significant. But in North America, for the most part, it's usually a pretty mild course. And then domestic sheep as well, they can get infected with blue tongue virus. And so blue tongue virus in domestic sheep can be a pretty significant disease problem for sheep.
You talked a little bit about it being insect vectored. Can we talk about that a little bit?
Sure. So Culicoides there's about 150 different species of Culicoides biting midges in the United States. And so Culicoides are, a vector, meaning they basically become infected themselves. So if you have a vector like a mosquito or a Culicoides biting midge, they will, they're blood feeding insects. So they will bite their host, take that blood up, and if those viruses are circulating in that animal's bloodstream, then that virus enters the insect's body and also results in an infection. And eventually, after a lot of activity in that little insect vector, gets back to their salivary gland. And then once they bite a new host, another animal, take another blood meal, they can transmit the infection that way. But
so they're not just mechanical vectors, there's actually replication in the insect.
Yes, correct.
I always think that's one of the cool tricks of viruses, to be able to replicate in two very different hosts.
It is. It's pretty wild. Yeah, when you really think about just how different those two creatures are, that virus has adapted to be able to utilize both sort of hosts. One's an insect vector host and one's a ruminant vertebrate host. But yeah, it's pretty unique. And these insects, they're very small, much smaller than a mosquito. So they're quite different than mosquitoes. Mosquitoes are basically like little hypodermic needles on wings, right? They're flying around. They put their mouth parts in the skin. They try to be stealthy. They try to put those mouth parts right into the vessel, right? They're just pulling blood out like a syringe. Culicoides are a lot more primitive. If you look at their mouth parts, they're basically just little oscillating saw blades. And so they're much more primitive feeders. So they're called what we call a laceration pool feeder. So they'll basically land on the skin. They'll just start biting with those little blades. And they're just trying to lacerate or nick a capillary, like some of the most small vessels that are in our body, and they're in our skin. So they basically just start biting until they nick a blood vessel. That blood starts to pool, and then they lap it up. And most people, if you've ever spent any time on the coast at the wrong time of year, you've probably been dined on by Culicoides biting midges. They're oppressive, and their bite is painful.
Now, are these the ones that are sometimes called no-see-ums?
Exactly.
Okay.
Yes. No-see-ums.
So small.
You can't see them. And then 5-0s, punkies are some of the common names for Culicoides biting midges or no-see-ums. So
yeah, I haven't spent a lot of time on the Georgia coast, but I think people have talked about some of these insects and not being in these places at the wrong time.
Yeah.
So I imagine that's what they're talking about.
Yeah. Side story. My first introduction to Culicoides biting midges, I was actually in high school. My sister went to school at Florida Southern University. So there was one time during high school, I flew down to see her and we drove down to the Keys and went camping. We went camping in August in Florida Keys. And it was beautiful. It was great. And then the first night, as soon as we got to that crepuscular time, like the sun starts to drop, the temperatures start to cool right when you want to be outside, we just got destroyed we couldn't see them. We didn't know what they were. It wasn't mosquitoes. And we were just a mess. We were jumping into the water, trying to escape these things. We would just get ourselves in the tent and just sit there and sweat, for hours until the sun went completely down and we could get back out. I didn't know what it was then, but I know what it was now. And that was my first introduction to Culicoides.
So coincidentally, my wife and I went camping in the summer in the Florida Keys and had the exact same. We camped for one night and then found a hotel.
Oh yeah. It was rough.
So listeners note, no camping in the Keys in August. But talking about the biting humans, do humans need to be worried about the diseases that these insects transmit?
No, luckily, epizootic hemorrhagic disease virus and blue tongue virus do not infect people. So it is not something we need to worry about.
So I know there's a lot of diseases that SCWDS focuses on, but there's one other disease of deer, chronic wasting disease, which I guess only recently have we found the first case in Georgia. So can you tell us something about that disease? And
yeah,
do we need to worry about it here in Georgia?
Yeah, I mean, it's nothing we need to panic about, but to some degree, everybody needs to be worried to the point of vigilance for chronic wasting disease. Chronic wasting disease it's a tricky one. It's probably one of the most, if not the most significant diseases of wildlife that state wildlife management agencies are facing. So those that are tasked with managing populations, it's certainly a challenge. There's so many reasons that these diseases challenge us, and it's a prion disease. So, most of the infectious diseases that we think about and are familiar with, we think about viruses, bacteria, parasites. These are things that have genetic material inside them that sort of help program their life history, tell them, what to do, how to do it. Prion diseases are literally just a protein. So these are abnormally shaped proteins that really are normally present in vertebrate bodies. We have normal prion proteins. A lot of times their exact function isn't always known, but these prions that have this sort of abnormal shape, they sort of resist the normal body's breakdown process. And so rather than go through the normal lifespan of a protein in the body where it might get broken down and those pieces are reused for other things, these proteins just stick around. And then they also, in some way, can sort of transfer that misfolded or misshapen form onto other prion proteins in the deer body. So they just accumulate over time. And so it takes a really long time for disease to occur in a white-tailed deer. And so that's one of the confusing pieces, I think, for a lot of people. Most of the infectious diseases that we think about, if I'm sure you guys talked quite a bit about COVID or influenza and some of these viral diseases that okay, if I came in sick today and I expose you guys, a handful of days, I got a sniffle, I got something in my throat. So it's kind of weird. You're going to feel that pretty quick. That's called the incubation period. With chronic wasting disease, unfortunately, this can be a year, 2 years, or more. So the time from when a deer actually gets exposed to that abnormal prion and it gets infected, it might be 18 months before somebody looks at that deer and says, that's a sick deer. And so one of the other problems is that during that period, they can actually shed via urine, feces, saliva. They can shed this prion into the environment and expose other deer, even though they look completely healthy and normal. The other problem, there's many problems, is that these prion proteins, once they're in the environment, they're environmentally persistent. So if that's a big curveball in the pathogen world, right? Creates a ton of problems when you're trying to manage a disease. And that's what wildlife agencies are trying to do. They're trying to manage this disease and prevent this disease. But, when the pathogen itself can just be pooped or peed or spit out and just hang out in the dirt and rocks and on vegetation and water and just sort of be there for years. We don't even know at which point it's not infectious anymore. So that creates another problem. So it's kind of one of those forever diseases, which is it's a significant problem. Once you have established chronic wasting disease in a population, it's more about finding ways to live with it than get rid of it, unfortunately. And so right now, this is very new occurrence in Georgia. There's been only one detection. It was a hunter-harvested animal in South Georgia, and there's only been one so far, right? And so in this time, it's basically trying to better understand what the situation on the ground is, right? So trying to get more samples from more deer in that immediate area, but also other areas nearby to try to figure out, okay, well, are we really early in this process? Did we luck out and get a very early outbreak starting, or is there more there that we just haven't found yet? It's very difficult to find the disease on the landscape. It's cryptic. It's very spotty. So
Is there a diagnostic where you can actually look for the presence of this misfolded protein?
So there's a couple different tests that we lean on heavily. Something called an enzyme-linked immunosorbent assay or an ELISA test, and then an immunohistochemistry or an IHC test. These are both sort of post-mortem samples, so like after death. And so what a lot of agencies lean heavily on are sort of hunter-harvested deer, so post-mortem samples. Or if there's a sick deer and it gets driven to SCWDS and we do a necropsy or a post-mortem exam on it, we're always testing those deer for chronic wasting disease. And so those are sort of the heavy hitter diagnostic tests that we have. And unfortunately, they're just post-mortem samples.
Yeah, you brought up a couple of things that I want to kind of touch on. The first is more of a comment than a question, which is I appreciate talking about things that are outside of the realm of the typical virus, right?
Oh, sure.
Bacteria.
Yeah.
And I'm glad we kind of are able to, in this platform, talk about things like prions, or we haven't really talked about spores would be another thing that's in the environment, and fungi, right? So these things that we don't talk about as much more mainstream that I really think we should talk about. That's more my comment. But my second is my question. You mentioned hunters. So does SCWDS have programs where they talk to hunters, they inform hunters of things to look for and how to report things back to y'all?
So that's a good question. So, with SCWDS, through our sort of membership with our state wildlife management agencies, we really serve them. And so we communicate daily directly with biologists, and many of our states now have veterinarians on staff too. So we're in constant communication with the biologists, and then those agencies, they have entire marketing, PR, outreach folks that sort of handle a lot of that direct communication with the public. So most of our direct communication is directly with wildlife biologists at state agencies, unless they ask us to engage or come to a public meeting or a legislative hearing and help with some of the communication about some of these complex diseases directly with the public or with policymakers.
So I know you've got a lot of different kind of research and projects going on at SCWDS, but there's one more I do want to touch on because I think it's very timely. And that is avian influenza. So you've had active research into avian influenza long before it was cool.
Oh, absolutely. That's a great way to put it. Yeah.
So tell us some about that.
Yeah. I'm pretty fortunate to be at SCWDS around some real global experts when it comes to avian influenza in wild birds. It doesn't get much better. And so we've had, Dave Stolnick. He has had a long-standing program with some funding by NIH and others, but really understanding low pathogenic avian influenza. That's not what we hear about in the news media. The news is talking about, when they're referring to bird flu, we're talking about highly pathogenic avian influenza. And so, the uncool part is the low path viruses and really understanding the ecology of the virus. And so how these viruses that are commonly occurring in waterfowl and shorebird populations for decades and decades, how they sort of persist in the environment and utilize hosts and natural history of the bird populations, migration. There's so much complexity there. And Dave and Becky, for many years now, they've, had such a good understanding of how these low pathogenic avian influenza viruses circulate in wild bird populations. It's been foundational knowledge for us now, addressing like a very significant need, both in terms of wildlife health, but also domestic animal health and to some degree human health. And so, it's that not studying something until it's cool, it's just a great way to put it, because it's been that knowledge that was built up over decades, today has become the beauty of doing all that work for so many years is definitely apparent these days.
I guess if you're avian influenza, whether low path or high path, waterfowl are basically your Delta Airlines.
Mm-hmm.
Yeah. I mean, that's the thing that I was going to bring up. I mean, it makes it so difficult, right? When you have something that's a moving target, and really a moving target.
Yeah.
And so does that mean that you have to also work cooperatively with a lot of other agencies as these birds are kind of moving around?
Yeah, absolutely. And Becky and Dave have an incredible sort of collaborative network it spans the country, but honestly the world to some degree. And so they've got some great long-term study sites, one in Delaware Bay that's kind of focused on shorebirds, and then some sites here in we kind of separate the country into flyways. So we've got the Pacific Flyway, the Central Flyway, Mississippi Flyway, Atlantic Flyway. These are mainly, the birds don't always mind these things, but they're general flight paths for like heavy migratory pathways. And so they've got some field sites sort of like for the Mississippi Flyway, for instance, up in Minnesota, where they'll sort of work with the wildlife agency. They'll catch birds, they'll sample birds, they'll band them. And then they also have sites in sort of Louisiana and Texas, at least in the United States, at sort of more of that southern end. So depending on the season, they're able to get their hands on birds, sample birds, and look for things like population immunity and antibody profiles in those birds, but also test them for actual viral infections. They've got some long-term work in Alaska. So if you look at sort of, that's the Pacific Flyway, and that's really coming from Russia and Eastern Hemisphere into Western. So it's like there's a lot of pathways that birds move. I mean, they're incredible athletes and long-term migratory species. The amount of movement that birds can do quickly is pretty astonishing. So it's a complex system.
In Alaska, that would be Andy Ramey.
Exactly. Yeah.
And was he trained here at SCWDS or at UGA?
Yes, he was. So yeah, Andy Ramey, he's at the Alaska Science Center, which is U.S. Geological Survey Center in Alaska. And because of the sort of the long-standing capacity and expertise that we've had here at UGA and SCWDS as it relates to flu, Andy did his PhD while still an employee at U.S. Geological Survey. So he came, it was probably around 2012-ish or so. But yeah, he did his PhD here at UGA and at SCWDS, and then went back to USGS. And so he is still there. So we've got Andy, a UGA grad up in Alaska, who's now just one of the core collaborators. That's pretty neat.
Cool. Very cool.
So I imagine we've kind of touched on this, that, you sit kind of at the forefront of frontline surveillance and diagnostics. And you hinted at this a little bit, but I want to kind of follow up on it. You talked about changes in policy or speaking to groups that maybe affect policy. So are members of SCWDS doing this type of work all the time?
Yeah. So in the seat that I find myself in now, this is kind of the realm I get put into a lot more than I guess I sort of ever imagined that I would, because at the end of the day, everybody has sort of some skin in the game, but often it's for different reasons, right? So the reason that I value white-tailed deer might be totally different from the way you value white-tailed deer. Mm-hmm. Does someone like to hunt it and harvest it? Does someone like to just watch it? Someone like to feed them? Are you a crop producer and you really don't like them?
I'm a gardener, so I can say that I don't like them in my garden.
Yeah, they will wreak havoc on your garden, right? And if you're a crop farmer, there's crop depredation, right? Where they're coming through and they're utilizing that as a food resource. They're just doing what deer do. They're taking advantage of resources on the landscape. They don't know any different. And then you have others where people put them inside fences and grow them for, financial gain or other reasons. And so you have all this diversity of interest in an iconic North American species. It tends to create conflict. And that example, that's just deer, that's the easy one, but there's plenty of other examples in the wildlife world where, conflict is created, whether it's between the agriculture and the wildlife sector or the private sector and wildlife sector, maybe human and wildlife health. So there's all kinds of reasons, but those are really sticky issues, and states often need some assistance to navigate some of those topics. And I come in usually on the disease side of things, obviously, because some of these disease systems they're just really complex and they're hard for science nerds to understand, let alone a politician who might be who knows what their background is, but it might be a completely different language that they speak than disease nerd language. And so just trying to help the agencies where they need some scientific expertise and some consultation and some backup to help explain certain things to people making decisions. Because at the end of the day, as scientists, we're great at making discoveries and making recommendations, but we're not the decision makers. And so it's that's a big gap sometimes. Sometimes it depends on the state or the situation or the region. Sometimes it's very easy, and sometimes it's a little bit more challenging.
Yeah.
Depends on the situation.
Well, I appreciate that perspective and also your desire to engage in that realm.
Yeah, it's not something I ever thought I was going to be doing, for sure.
Well, our guest today is Dr. Mark Reuter, faculty in the Department of Population Health and director of SCWDS, the Southeastern Cooperative Wildlife Disease Study. Mark, thanks for being with us.
Yeah, it was a pleasure. Thanks for having me.
Links to additional information about Dr. Reuter's research can be found on our Instagram account, which is at PPPPodcastUGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched this season. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Special thanks to our production assistant, Sid Wiggin, for research, editing, scheduling, and keeping us on track.